Hi everyone. Before we actually start to talk about deep learning models applied to molecules, let us look at the question, why is it even interesting or necessary to make predictions for different kinds of molecules? At the most fundamental level, life is composed of various types of molecules that interact in complicated ways to create biological structures and functions. The, these molecules, for example, include larger molecules such as DNA or proteins, which are often described as molecular machineries, or smaller molecules such as simple sugars, which can serve as energy sources. You can see a visualization of a protein and of a small molecule here on the right side. The different balls that you see in these images are the atoms of which the molecules are composed of. If you don't have a large biological or chemical background, don't be afraid. In future videos, I will explain to you everything that you need to know about molecules to be able to follow the content. So, why do we want to predict something about molecules? Understanding the function of molecules is crucial for many different scientific fields, including medicine, biology, biochemistry, which is the study of chemical processes within and related to living organisms, or environmental sciences, for example, when studying and managing pollution. One example from the field of medicine where it is important to understand properties of molecules could be the following. If you want to treat a disease and develop a drug for it, we often need to search for molecules that either inhibit or activate the function of a protein. Therefore, it could be beneficial to be able to know which molecules interact with a certain protein and also to know in which way they interact. For example, do they activate or inhibit the protein's function? Another example from the field of biology could be understanding the metabolism of cells. How can a cell grow and produce all the relevant molecules that are required for cell growth? To properly understand this, we need to determine the functions of many different molecules. On the other hand, molecules often play an important role in industrial settings. For example, you could be interested in finding a material which should have some specific properties, such as high strength or specific electrical characteristics, which depends on the molecular structure and the molecule composition. You could use prediction models to screen a large number of different molecules for a property of interest without the requirement of executing an experiment for each of those molecules. And another example could be that you're interested in generating a specific molecule starting from a different molecule. For example, you could be interested in producing a molecule named 2,4-dihydroxybutyric acid, or short DHB, starting from malate. The DHB molecule is important for the production of many pharmaceuticals, but its natural abundance is low. On the other hand, the natural abundance of malate is high, and it is thus highly desirable to find proteins that can convert the malate molecule into the desired target molecule. To achieve this, you need to find proteins with specific functions, and in this case, it is actually possible to use 3 different proteins and 2 intermediate molecules to produce DHB molecules starting from malate. On this slide, I want to make clear why it might be necessary to develop machine learning models for predicting molecule properties. So, what are the different methods that can help us to determine properties of molecules? An obvious method is to directly measure molecular properties using experimental techniques. For example, if we would like to know if a given protein catalyzes a specific chemical reaction, which means we want to find out if a given protein can convert a starting molecule into another molecule of interest, we could experimentally measure if this protein converts the starting molecule into the target molecule. The advantage of direct experimental measurements is that the results are usually more accurate than alternative approaches. However, the disadvantages are that it can be very time consuming, difficult, and expensive to perform such experiments. For example, if you want to measure if a protein is catalyzing a specific reaction, you will first need to produce and purify the protein, get a concentration of your starting molecule, and then perform the experiment. Especially if you want to test your protein against many different molecules, this can take a lot of time. An alternative to experimental methods are non-machine learning computational methods that simulate the behavior of molecules using theoretical principles, empirical data, and statistical analysis. Advantages of these methods are that you usually get your results faster at reduced cost, and the methods are usually much more accessible and easier to execute than experiments. However, disadvantages are that often experimental results are more accurate than computational methods, or it might be infeasible to simulate very complex interactions, and you might require large amounts of high quality experimental data to develop these tools. In these cases, it can be better to perform a single experimental measurement. And the last alternative is the one this course is concerned with, machine learning prediction models. That means using data, usually experimental data, to train a model to predict a certain property or activity of a molecule so that we can make predictions when no experimental data is available. The advantage of this approach is that machine learning models are capable of handling highly complex data and also, for example, handling complex interactions between molecules. Machine learning models are excellent at detecting patterns in data that we might not be able to find without such models. Once machine learning models are trained, they are much quicker than experimental methods, and they can be flexibly retrained and readjusted once more data becomes available. To come back to our example from the experiments, if you want to find out if and which molecule a certain protein can catalyze, experimentally testing the protein against a large number of different molecules can be very time consuming and costly. And also, traditional computational methods are also not very useful for the task, as the interaction of a protein with other molecules is very complex, which we cannot easily simulate. Instead, we could train a machine learning model for this task. We could use lots of available experimental data for other proteins where we know with which small molecules the proteins interact, and train a model to predict protein small molecule interactions. Using such a model would allow to predict promising candidate molecules that the protein can catalyze. This would allow us to only test a small number of promising molecules experimentally instead of making experiments for a large number of different molecules. The disadvantages of prediction models include that typically the accuracy is lower compared to experimental methods, and we usually need a lot of data to train successful prediction models. Machine learning models can also be hard to interpret, which means that it can be difficult to find out why the model made a certain prediction. And finally, related to the accuracy, most machine learning models are prone to overfitting, which means we have to be very careful that the prediction models do not only remember the training data and the noise in the training data, but also generalize well to data points outside of the training data. So in summary, machine learning models are very useful if traditional computational methods cannot solve the prediction task successfully, or experiments are too costly and or time consuming, as in the example of predicting protein small molecule interactions. If sufficient experimental data is available, machine learning models are very good at extracting patterns and useful information from the underlying data. Now that we know that it can be really beneficial to be able to predict properties of molecules, we will have a look at a few more specific prediction tasks to get a better idea of what kind of properties can be predicted using machine and deep learning models. Let's, for example, take a protein which has a specific function. This protein will be most efficient in a specific environment, for example, at a specific temperature and a specific pH value. if we are interested in using a protein for an industrial purpose, we want it to be as efficient and stable as possible, and we need to find the optimal conditions. We could measure experimentally how efficient the protein is at different pH values, which tells us how high the concentration of hydrogen atoms in the environment is. But doing this experimentally can be expensive and time consuming. Alternatively, we could build a prediction model that predicts the optimal pH for this protein by using information about the protein. An important part of this lecture will be to find good ways to extract relevant information about the molecule, in this case the protein, which we can then use to make a prediction about a specific property. Something else that we could be interested in could be predicting the effect of small changes to the protein, so-called mutations. For example, we could be interested in changing our protein a little bit to achieve a protein that is optimal under different conditions, but still has the same function. To achieve this, we could build a prediction model that predicts the effect of a certain mutation. And of course, these are just two out of many examples for what we can predict for proteins. On the other hand, we could also be interested in predicting a property of a small molecule. When developing a new drug for treating a certain disease, this drug should have as little as possible toxic effects. How toxic a small molecule is means in this context the degree to which it can damage an organism. Instead of performing costly and maybe harmful experiments, we could also use a machine learning model to learn from existing data to predict how toxic a certain molecule will likely be in the human body. An important part of this process is similar as for the protein property prediction task, to extract meaningful information from the small molecule that we can use to make a prediction for the small molecule. We will later see how we can use deep learning methods to achieve this. Finally, it can also be the case that we are interested in not only predicting a property of a single molecule, but we could be instead interested in predicting how two molecules interact with each other. For example, a protein and a small molecule. Enzymes are proteins that bind to small molecules and transform these into different small molecules, the so-called products. And hence, we could be interested in predicting, given a protein and a small molecule, does the protein bind to the small molecule? Another example is predicting how small or how much a small molecule inhibits the function of a protein. When developing a drug, we are often interested in finding small molecules that inhibit the function of a certain protein. So we could be interested in predicting how much does a specific small molecule inhibit the function of a protein. Now that we know that it is very beneficial for a vast amount of different industrial and research areas if we can make predictions for properties and functions of molecules, we will have a look at what kind of different molecules we will mostly cover in this course. We will focus on biomolecules, which are molecules that are present within and produced by living organisms. They are essential for life as they serve as the building blocks of cells and organs, carry genetic information, and perform various functional roles in metabolism and regulation. However, although we will mostly focus on biomolecules, the methods that we will cover are in principle applicable to any kind of molecule. We can broadly divide biomolecules into two different categories: micromolecules and macromolecules. Micromolecules are small, simple molecules with low molecular weight. They are important as building blocks for the synthesis of macromolecules or as energy sources and signaling molecules. Examples of micromolecules include vitamins, amino acids, monosaccharides, which are small sugars, nucleotides, and fatty acids. Macromolecules, on the other hand, are large complex molecules with high molecular weight. Examples of macromolecules include proteins, nucleic acids such as DNA and RNA, and polysaccharides, which are large sugars. The distinction between macro and micromolecules is generally based on molecular size and weight, as well as complexity and functionality. However, there is no strict cutoff point in terms of molecular weight or size that universally applies to classify a molecule as either a macro or micromolecule. The classification often depends on the context and specific characteristics of the molecule of interest. In the first part of this lecture, we will mostly be interested in making predictions for proteins, which are the molecular machineries of all living organisms, which means they perform a wide variety of functions critical to the structure, function, and regulation of organisms. Later, we will also investigate how we can make predictions for all different kinds of small molecules.